Decoding the epigenetic clock of aging. How old are you really? Start with an engaging question. What if I told you that the age on your birth certificate isn't the full story? You might be 50 years old according to the calendar, but your body could be functioning like a 40-year-old's, or vice versa. This isn't science fiction. It's the critical difference between chronological age, the number of candles on your birthday cake, and biological age, the true age of your cells. Over the last decade, scientists have discovered an incredible tool to measure this biological age, the epigenetic clock. This discovery is completely changing how we understand aging, transforming it from an unavoidable destiny into a process we can measure, understand, and maybe even influence. Part 1. The software of life. What is epigenetics? To understand epigenetic clocks, we first need to talk about epigenetics. Imagine your DNA is a giant cookbook with thousands of recipes, genes, for everything your body needs to function. But not every recipe is used at the same time. A skin cell doesn't need the recipe for a liver cell. Epigenetics, which means above genetics, acts like a master chef. It doesn't change the recipes, your DNA, but it decides which recipes get read, when, and how much. It does this by adding tiny chemical tags to your DNA that act like on-off switches for genes. The most studied mechanism is called DNA methylation, and it's the foundation of the epigenetic clock. Your epigenome isn't fixed, it's dynamic and can change based on your lifestyle, your diet, exercise, stress, and environmental exposures. This explains why genetics aren't destiny and why our choices have such a profound impact on our health. Part 2. The aha moment, the birth of the clock. The story of the epigenetic clock begins with an accidental discovery by a UCLA scientist named Steve Horvath. For years, he had tried to create an aging clock using other methods without success. Then, in 2011, while analyzing DNA methylation data for a completely different study, he noticed something incredible. The patterns of these chemical tags could predict a person's age with frightening accuracy. After years of data collection, in 2013, Horvath published the first pan-tissue epigenetic clock. It uses 353 specific sites on the DNA to predict chronological age with an average error of just 3.6 years. The most amazing part was that it worked across almost every tissue in the body, blood, brain, skin, using the same algorithm. This suggested that aging isn't just random wear and tear. There's a fundamental systematic program running in the background. Part 3. Not all clocks are created equal. The evolution of accuracy. The first clocks were great at guessing your calendar age, but they didn't tell you much about your health. This led to the development of second-generation clocks, which were a game changer. Instead of predicting age, these new clocks were designed to predict health outcomes. The two most famous are 1. PhenoAge. This clock incorporates clinical biomarkers, like creatinine and C-reactive protein, to create a phenotypic age that captures your overall physiological state. It's a much better predictor of disease and physical function. 2. GrimAge. Often called the Grim Reaper of clocks, GrimAge is the most powerful predictor of time to death, cancer, and heart disease. It was built by predicting the levels of death-related proteins in the blood and even a person's smoking history, all just from their DNA. Third and fourth generation clocks are even more advanced. Dunedin pace acts like a speedometer, measuring your current pace of aging, not just a static odometer. And the newest clocks, like Damage, are trying to separate harmful changes from adaptive ones, giving a more detailed look at why some people age healthily and others don't. Part 4. The big question. Is aging the cause or just a symptom? This is the central debate in the field. Do the epigenetic changes the clocks measure actually cause aging, or are they just a symptom of something deeper? The passenger hypothesis. Some scientists believe the root cause of aging is the accumulation of random mutations in our DNA. In this view, the epigenetic clock is just tracking this damage. Reversing the clock would be like treating a fever without curing the infection. The driver hypothesis. Others argue that the epigenetic changes themselves are the primary driver, disrupting our cellular programs and causing functional decline. Recent research has revealed a surprising link. DNA mutations appear to cause a predictable cascade of epigenetic changes. This suggests that reversing aging might be much harder than simply resetting the epigenetic tags. If mutations are the root cause, then fighting aging may require addressing both the hardware, DNA, and the software, epigenetics. Part 5. Can we turn back time? This is the billion-dollar question. And the surprising answer seems to be, to some extent, yes. A landmark 2019 study called the TRIEM trial showed that a cocktail of 3 drugs reversed the epigenetic age of participants by an average of 2.5 years after 1 year of treatment. While it was a small study, it was the first proof of concept that reversing the biological clock in humans is possible. More importantly, for most of us, lifestyle choices have a measurable impact. Studies show that. Diet. A diet rich in vegetables and fruits, like the Mediterranean diet, is associated with a younger epigenetic age. Exercise and sleep. Regular exercise and sufficient sleep also have a positive effect. Negative factors. Obesity, smoking, and chronic stress all accelerate the clock. Part 6. The promise and the peril. This technology has enormous potential to revolutionize medicine. Imagine identifying people at high risk for Alzheimer's or heart disease decades before symptoms appear, allowing for preventive interventions. The clocks could also dramatically speed up the development of anti-aging drugs by providing a quick way to measure if a therapy is working. However, there are significant challenges and ethical concerns. Accuracy and bias. Most clocks were developed using data from European populations. They may be inaccurate for other ethnic groups, potentially worsening health disparities. Also, a clock trained on blood might not work well on brain tissue. Discrimination. The biggest fear is that insurance companies or employers could use biological age to make decisions, creating a new form of biological discrimination. Could you be denied insurance or a job because your clock ticks a little faster? These are serious societal questions we need to address. Conclusion: you are the architect. The epigenetic clock has changed the conversation about aging. It's no longer a passive process that happens to us, but a process that is measurable, influenceable, and has reversible components. While this technology is still in its early days, it offers an incredible promise of a future where medicine focuses on maintaining health instead of just treating disease. It also empowers us, showing that our daily choices are literally writing the story of our health onto our very DNA. The clock is ticking for all of us, but for the first time in history, we're learning how to read it. And maybe, just maybe, we're learning how to adjust its speed. Subscribe for more tips on decoding the epigenetic clock of aging. Like, comment your favorite peaceful practice, turn on notifications for weekly inspiration. Thank you for watching.